is Anna Blümel. I'm a second-year PhD student in the Royal College of Surgeons in Ireland. My work is funded by the Science Foundation Ireland and it is based on work from Breast Predict, an Irish Cancer Society funded collaborative research project. In this video, I would like to show you what my research is about and why what I do matters. The aim of my PhD is to guide clinicians' decision when treating a subgroup of breast cancer patients called HER2. I'm doing this by deciphering the signals we get from breast cancer cells. However, deciphering these signals often feels like talking to aliens, which is why I would like to introduce you to 3 of them. I have a massive issue in understanding them. All of them seem to be unwell, and I have decided to do something about it, so I give them some tea. 2 of them look happy now, but the other one still unwell. What should I do next time an alien is sad? Should I give them tea or not? As I said, my research focuses on understanding the signs of an unknown species, so I did some research and I tracked what signals the aliens gave me. Here are my notes. I have kept track of the number of eyes they have, if they roll their eyes, the words they use and their reaction to tea. In this case, it seems like the rolling of the eyes is a signal that they need tea. The unknown species I'm trying to understand are breast cancer cells. I'll show you what they have in common with aliens. Just like aliens, they are very different from each other in appearance and behavior. Their reaction to treatment is often as uncertain as the reaction of aliens to tea. What is certain, however, is that treatment is expensive and causes severe side effects. Therefore, we often ask the question, should we give the patient the treatment or not? As a result, there is a huge need to get rid of this uncertainty and to tailor the treatment to every patient. Fortunately, we know that the reaction of the cancer cell to treatment is often linked to certain signals, similar to the reactions of aliens to tea and the rolling of their eyes. The most widely known signal is the cell's DNA. The cell gives us a huge number of other signals, such as RNA and DNA methylation, which can control the DNA activity and therefore the gene function. In the research group that I'm part of, we characterized these 3 types of signals for real breast cancer tumors, along with the patient's reaction to a drug called lapatinib. This drug is given to breast cancer patients whose cancer cells make too much of a protein called HER2. The data I have for these patients is somehow similar to my notes about the aliens. However, my notes about the tumor samples are on average 30 gigabytes per patient. This is way more than I had about the aliens. If I printed that data for 1 patient on normal paper, normal size, the weight of that paper would be the weight of 30 cars. The aim of my PhD is to make sense of that data. More precisely, I aim to find a way to predict patients' reaction to lapatinib with patterns in that data. Say, for 1 patient, we find a certain pattern in the data shows that the patient will most likely benefit from taking lapatinib, as it will kill the cancer cells. This patient should get the treatment, even though it comes with costs and side effects. If this pattern is not present for another patient, we know that the patient's cancer cells would not respond the same way, and the patient would have the same outcome with or without taking lapatinib. We should search for different treatment for this patient and avoid the unnecessary costs and side effects of lapatinib. So, what if I find something? Well, 1 in 8 women develop breast cancer in their lives. Approximately 20% of all breast cancer patients are HER2 positive and could potentially be treated with lapatinib. In conclusion, this means that for 1 in 40 women, a clinician will at some point have to decide if that woman would benefit from lapatinib treatment or not. This was the case for more than 400,000 newly diagnosed women only in 2018. If my research is able to make prediction about the patient's reaction to lapatinib, it could potentially be used to tailor the treatment for every one of these women. This could reduce treatment costs and, more important, avoid unnecessary side effects. Thank you very much for watching this video. I hope you see why my research matters.